Hey everybody, happy Earth Day. 50th anniversary, I guess. That's kind of cool. So anywho, here's what we're doing. You guys did this quiz last time and you got whatever score you got, but I'm going to go over the answers with you guys carefully because the important thing is to learn how to be successful on this test so that you can earn your certification. So this is just a practice test. This is version A of the practice test. I'm going to go through. So I'll assign you the first quiz. Like for example, here's all the quizzes: quiz 1, quiz 2, quiz 3, 4, 5, 6, 7, 8, 9, like that. And then each day I'll assign you one of those, and then the next day I'll make one of these videos and go over the answers with you one by one to make sure that you're understanding it well. You're going to get a score for that, but I'm not going to give you like a grade based on the score. I just want you guys to do it and try your best. This next one, this version B, we'll do the same thing, except what I'm going to do for that one is I'm just going to have you do the whole thing and get your score. I might have you put a little skin in the game on that one as far as a grade goes. I'm not sure yet. We'll see. But either way, I'll hold like a online Zoom thing or whatever, and you guys can ask me questions and I'll answer them. And then the third time is going to be the real go, which is going to be the real test. So anywho, let's go over this this first one. So question 1, match the compound to its corresponding bond type. Let's see what our choices are: ionic, nonpolar, covalent, and polar covalent. Ionic is where electrons are stripped away and you end up with ions that stick to each other. Covalent is where they're shared. Nonpolar means they're shared perfectly. Polar means they're sharing, but one of the atoms is hogging them more than the other. So sodium chloride, we got a metal and a nonmetal. That's ionic. Water is the poster boy for polar covalent. I mean, we just know this. Methane is a hydrocarbon. So you guys know that hydrocarbons are non-polar, and that's why hydrocarbons and water do not mix. They're not miscible. So methane would be non-polar covalent. Question 2, which of the following should be used to calculate the amount of a compound required to make a solution? I actually got this wrong when I did it because I didn't read this right. I thought it said which of the following should be used to measure the amount of compound required, and I chose empirical, and empirical means you saw it with your own eyes, and I got it wrong because I didn't read the question. It says calculate, and in order to do the calculation you need to turn from the molar mass into grams so that you can use the scale or the balance. So that would be the molecular weight that you would need to use. Question 3. Below is a list of 4 groups of macromolecules. Macromolecule means a big ass molecule found in living things. Match the macromolecule to its example and associated functions. So carbohydrates, lipids, nucleic acids, proteins. You might be like, carbohydrates, I know what that is. That's like pasta and stuff. Lipids, you might not even know what that is. It's okay, man. It's all about your test taking skills. It's not about what you know. So carbohydrates. Here's our examples: enzymes, cellulose, energy storage, cholesterol. We don't know what that is. Membrane structure. So anyway, when I see energy storage right here, and you might know that cellulose is another name for wood. We're going to know that that is the carbohydrate. Lipid, maybe I'm scared of that word. I don't know what it is. I'm going to come back to it. Good test taking strategy. Nucleic acids, that sounds like nucleotide, which sounds like DNA to me. So I'm going to come down here and find something that's kind of like DNA, RNA. Booyah. And then proteins, we know in this class, you better know that those are made out of amino acids and they fold into shapes that do things. So we're going to look for something like that. Enzymes, we know, are proteins. Structures, receptors, transports, boom. So we're just looking for keywords that are going to tune us in. And now Let's see if the last thing we have left makes sense for lipids. You might know that lipids are fats, and you might know that cell membranes are made out of lipids, like phospholipid bilayer, you know? So anyway, lipids, we're going to look for something that looks like fats. Cholesterol, that doesn't look like fats, but membrane structure. Membrane as in cell membrane. And energy storage, we know that you can store a buttload of energy in fat like a big fat whale. So booyah. There we go. Yeah? Okay, question 4. A solvent is the dissolved matter in a solution. Sometimes I won't even look at the possible answers. I just put my hand up over the screen so I couldn't see them. I'll try to figure out the answer by myself before I look at the possible answers. So I'll look at that and I'll be like, no, it isn't. A solvent is the thing that's dissolving the matter. It's not the dissolved matter. The dissolved matter is the solute. So this is crap. Boom. Question 5. Carbon dioxide as a byproduct benefits the brewing and baking industry in a process called fermentation. When microorganisms, usually yeast, undergo fermentation, they get their energy by this or this or this or this. So this is going to be a little sketchy for you guys, but this was the classic example that was used in biology to compare and contrast aerobic versus anaerobic respiration. Aerobic means with air, anaerobic means without air. Both of them produce carbon dioxide, but one of them requires air and one of them doesn't. And when yeast are inside of a beer bottle making beer, or deep inside like a lump of dough, they run out of air and they switch from aerobic respiration to anaerobic respiration. So both of these suckers are actually kind of right, but this is the better answer, anaerobic respiration. Okay, this question right here, you don't have to know you don't even have to know what glucose is or maltose. It doesn't matter. This is a graph reading question. This is studying your graph reading skills. So when you see stuff like this and you see these things that might tend to freak you out, just relax. All they're asking you to do is read a graph. The enzyme maltase catalyzes, that means speeds up, the breakdown of maltose into glucose. Maltose is turning into glucose. A technician adds maltase to a solution of maltose. Maltase is the enzyme and maltose is the sugar, to be incubated at 37°C for 20 minutes. Samples were taken every 5 minutes and graphed blah, blah, blah. Which of the following graphs represents the change in glucose and maltose? Looks like the technician put a bunch of maltose in there. And maltase enzyme breaks down maltose and turns it into glucose. So what should the concentration of maltose be doing over time? That's right. It should be going down. And what should happen to the concentration of glucose over time? That's right. It should be going up. So all we need to do is look for a graph where the concentration of maltose is going down and the concentration of glucose is going up. And that's happening in this graph up to this point right here. But then glucose starts to go down for some reason. And why would that happen? So let's see if we can find a better one. Here we got maltose going down and then flatlining, and glucose going up and flatlining. That makes sense. Let's see if we can find a better one. Here we got both of them going up. Crap. Here we've got glucose going down and maltose going up, which is backwards of what should be happening. So the best answer is this one right here. Boom. Question 7. The enzyme maltase catalyzes, speeds up the breakdown of maltose into glucose. Same thing. Technician does the same thing, and they're supposed to be incubated at 37°C, but something funny happens and the temperature of the stinkubator or the water bath doesn't get up to 37°C. It goes to 25. Well, that protocol called for 37°C, because that's the optimal temperature for this enzyme. So any temperature different than 37C is going to decrease the activity of the enzyme. So what that means is it's going to slow the activity of the enzyme down. It might not totally stop it, it may or may not, but it's definitely going to slow it down. So we're going to look for the best answer there. The enzyme activity would increase. I'm going to stop reading. That's not going to happen. The enzyme activity would decrease. That's going to happen, yielding the same level of glucose over a longer time period. It's possible. It could be that if the enzyme's working slower, if you give it more time, it'll still get all the work done. It'll just take more time. So that's possible. The enzyme would become inactive, yielding no glucose. That's definitely a possibility. The enzyme activity would decrease, yielding more glucose over a shorter period of time. That doesn't make sense. If the enzyme activity decreased, it would yield less enzyme. So it's between these two answers right here, and to tell you the truth, you guys wouldn't know. I mean, if the enzyme's going to work at room temperature, 25C is like warmer than room temperature. So it's still going to work. It's just not going to work as good. So this is the best answer. How would you know that? You wouldn't, but guess what? Now you do. How does DNA differ from one species to another? The way the nitrogenous bases pair together. That's crap, because A always goes with T and C always goes with G. The type of nitrogenous bases the DNA contains. That could be read different ways, and I think this question wasn't written very well. Maybe the order in which the nitrogenous bases are assembled, that would be obvious, but the type, I'm going to say no, because they all have adenine and thymine and guanine and cytosine. They all have the same 4 nitrogenous bases. The directionality of the DNA strands. It's always 3' to 5' on one side and 5' to 3 on the other. The number of genes and non-coding regions. Yeah, this is a possible answer, and it's the best answer in this crappy question. Different organisms have different numbers of genes, and they have different segments of DNA that does not code for genes. So now you know. Question 9, match the type of DNA to its correct description. Circular, long DNA containing several thousand genes. That's a buttload of genes, but it's circular. We know that eukaryotes, which we are, do not have circular DNA. We have linear chromosomes. So it's not going to be eukaryotic. It's either going to be prokaryotic DNA or plasmid DNA. But you guys know that you can't stuff several thousand genes into a plasmid. Plasmids are way too small for that. So it must be prokaryotic DNA. And prokaryotic means bacteria. Circular is the key word there. Cytoplasmic circular DNA containing only a few genes. Circular, key, and few genes, key. That means they're talking about plasmids. Linear, long, with many thousands of genes contained within a nucleus. Booyah! Only eukaryotes have nuclei. Okay, here we are. A structural genes code is blah, blah, blah. What would the messenger RNA transcript be? We know that T goes with A, so our first one's going to be A, and look, they screwed us. We can't eliminate any of those because they all go to A. The next one, A goes with T usually, but there ain't no T in RNA. Instead, there's U, so our next one's going to be U, so it's going to go AU. So now it's going to be one of these 3. So at this point, I'm going to look here, and they're all the same until this one right here. So let's see if that 4th one is, nope, it's not this one. So it's either this one or this one. And here's the big difference between these, GGG and CCC. So if I go up here, if this is CCC, then this is going to be GGG. So booyah. It's going to be that one. You can just write it out on paper and make yourself safe and don't even worry about it. The three-dimensional structure of a protein is largely determined by the amino acid sequence. You bet your bippy it is. A single base pair change in a gene's DNA sequence can affect the shape and function of the protein it encodes. A single base pair? Yeah, that's right. That's called a single nucleotide polymorphism, and lots of times it doesn't make a difference, but sometimes it does. We studied in biology that sickle cell anemia happens due to a single nucleotide polymorphism. We studied this year that the taster gene for that, dang, we never even got to finish that lab. That's crap. The taster gene for tasting that PCT paper is due to a single nucleotide polymorphism. True. All right. This one, I hope you guys freaked out at first and then realized that it's kindergarten. Match the protein structure to its level of structure. I never taught you guys this explicitly, but like I said, this isn't a knowledge exam so much as it is a test taking skills exam, and you guys are going to get good at taking tests. So anyway, what kind of choices do we have here? Let's see. Primary, secondary, tertiary, quaternary. What's that mean? I don't know, but I do know that primary means first and secondary means second and tertiary means third, and I can assume that quaternary means fourth. So we're looking at levels of structure from first to fourth. So what's the most basic structure here? That would be this structure right here, which is a string of amino acids, which we know is called a polypeptide. And that's what comes out of the ribosome's butt. Boom. We know that that comes first. Next, we're going to look at these things and we're like, crap, I don't know. But if we look right here, we see that this structure contains something called a pleated sheet and something called an alpha helix. And if we look here, we can see that this is an alpha helix and this is a pleated sheet. So we know that this contains both of these. So we know that this is more of a fundamental structure than that other one. I'll leave this one, but I'll show you what's up with that one in a second. So that would mean that that one was secondary and this one was tertiary or third. And if you look at this structure of this globby thing right here, look at that. It's the same as this right here, but this is connected to another globby thing. So what this is a polyprotein. This is a protein made of two proteins, kind of, or two subunits at least. Like hemoglobin is a polyprotein made of 4 protein subunits. So anyway, this is what's called quaternary structure right here. Booyah. Question 14. Select the option that represents the correct order of assembly in the cell. Which order or is whatever they're talking about getting assembled, first, second, third, whatever. So we got polypeptide, amino acid, protein. We know polypeptides are made of amino acids, so that's crap. Amino acid, protein, polypeptide. We know that proteins are made of polypeptides, so that's crap. Protein, crap. Amino acid sounds good. Polypeptide sounds good because polypeptides are made of amino acids, and then protein looks good because proteins are made from polypeptides. Boom, like that. See how we did? I bet we did good. I don't even know if it's going to score it because it's just a preview thing, but whatever. You guys will take my... Oh, did we get something wrong? Correct. Oh, that's just my score from before when I got that other one wrong. Yeah, we got all these right. So everything's cool. Everything's cool. Everything's cool. I miss you guys. I'll see you guys later. I'll see you one day. Okay, bye.